All right, we'll just get started in respect of time. Again, thank you for everyone joining us on our webinar on Introduction to ABC Atlas. This is the last webinar in a series, and we're really excited to have you guys here. So reminder, this webinar is being recorded. If you're watching a recording on this, it's on our YouTube channel, or if you're watching live, go to our YouTube channel. This actually webinar is part of a series. It's the last of our series. So if you are wanting to see a previous webinar, they're all on our YouTube channel at Allen Institute. This webinar is also kind of a different webinar if you've seen our previous ones. This one is really focused on Introduction to ABC Atlas, but also we're trying to hear from you guys. What feedback or resources do you need to help better your research with ABC Atlas? So we'll have questions throughout this webinar. If you're watching a recording of this, the same questions are in a survey in the description. And we really encourage you guys to fill that out. And as a disclaimer, this webinar, all the information we're showing is as of May 2024. So if there's a little bit changes or new data sets, this is just as ABC Atlas is as in 2024. So today's agenda, we'll kind of go review what type of data is in ABC Atlas in a very broad scope. Then we'll talk about what data sets are in ABC Atlas, what the ABC Atlas is and how to use it, some use case examples, the supporting resources we have here at the Allen Institute for the ABC Atlas, and then also asking you guys, what would you like to see along with a Q&A? Again, as I said, we really want this webinar to hear about you guys, about what you need to help your use of ABC Atlas. And with that, I'll kick it off to Rachel to talk about transcriptomic data.
All right. Hi, everyone. I'm Rachel Hostetler. I'm a scientist here at the Allen Institute for Brain Science. And my role at the Institute really focuses on providing technical support and training to scientists who are using our taxonomy tools. So if you have any questions after this webinar, if you're confused, if you just want some one-on-one support, feel free to contact me and we can set up a way to help you understand the ABC Atlas better. And so as a background of what the ABC Atlas is built on, so the ABC Atlas is a tool specifically that features transcriptomic data primarily from single-cell RNA sequencing data and also spatial transcriptomic data from MERFISH experiments. So first, I'm just going to give a broad overview of what these terms mean and what type of data we have here. So what exactly is transcriptomics? So transcriptomics is the study of RNA expression, also called the transcriptome, and how it differs between cells, tissues, and conditions. So when we talk about the genome, we're looking at our genes. When we're looking at the transcriptome, we're looking at the RNA. And when you look at the proteome, we're looking at our proteins. But all of the data collected here today is from RNA sequencing. So it's really looking at where the RNA is. And just a general overview of how single-cell RNA sequencing works. So first, you have your tissue of interest. The data featured in the ABC Atlas is brain, so brain tissue. After you have your brain tissue, you have to lyse the cells and extract your mRNA. Then from your mRNA fragments, you need to generate a cDNA library. Once you have your library, then you can sequence it, align your sequencing, and then analyze your data. So this is just a very broad overview of how this works. And depending on which technique you're using for your single cell or single nucleus RNA sequencing, these steps can vary a little bit just depending on equipment. But overall, this is a general overview of how this type of data is collected. And another type of RNA sequencing is called bulk RNA sequencing. And with bulk, all the tissue stays together and you actually get a mixture of RNA and it's your RNA expressions between all of the cells together. And then the newer technology that I just described is called single-cell RNA sequencing, where you actually get to keep your cells or nuclei separated in your experiment, which allows you to look at the RNAs and their expression at an individual level. So bulk RNA sequencing has been around for longer. It's great for looking at tissue, and if you don't need to know your single-cell identity. But the data featured in these data sets is at the single cell level, where we're able to actually isolate the cells and say, oh, this RNA belongs to this cell, this RNA belongs to this other cell. And so an analogy that is often used in the transcriptomic field on how to describe these techniques is that bulk RNA sequencing is kind of like a smoothie. You have all these ingredients, but they get mixed together, and by the end product, you lose your identity of your individual fruits. But with single cell or single nucleus RNA sequencing, you actually keep the identity of those individual cells, much like this fruit basket here. which is really useful for looking at heterogeneity in a population. And what's the result? What's the output of this sequencing data? Well, what you end up getting is a cell by gene matrix, which shows you the number of transcripts counted for your different genes and also per cell. So when you have this over the course of thousands and thousands of cells and thousands of genes, this ends up being a matrix that can possibly have, 30,000 genes and however many cells that you profile. So it ends up being this very, very large data set. And this is highly dimensional data, right? Because you have one cell, and if you have 30,000 different genes there, that's a lot of information. Then you have your next cell, and you also have those same genes, but they're expressed at different levels, and then so on for all the cells that you've expressed, that you've profiled. And so these matrices end up being really large, and it's not always easy to graphically represent them. So a lot of times different dimensionality reduction techniques such as UMaps or T-SNEs are used to show this really highly dimensional data in a 2D space. And I'll get more into that in the next couple of slides. And a few differences, because this often comes up when talking about transcriptomic. There are some differences between whether or not you're collecting information from single cells and you're doing single cell RNA sequencing or single nucleus. So depending on the type of tissue that you're using, depending on if that tissue was fresh, if it was frozen, different techniques work in different ways. And they also profile slightly different areas, right? Your single nucleus is only looking at nuclear transcripts, whereas if you're doing single cell, you're also getting transcripts that are in the cytoplasm. And so for the human data that you'll be seeing in this presentation today, the human data will be single nucleus, just based on the limitations of the technique. Single cell data, single cell RNA sequencing doesn't really work with frozen tissue. A lot of the mouse data is single cell RNA sequence data that came from fresh mouse tissue. And this is something that scientists at the Allen Institute have been looking into a lot, the differences in quality between single cell and single nucleus data. Here's actually a paper from 2018 where a group of them actually went and compared the differences and how comparable they are. And what they have found is that both single cell and single nucleus can be used to identify and profile cells. They just are slightly different techniques, and you do get slightly different results. But if you're really interested in looking at the differences between these techniques, I would check out this paper here that I just listed below.
So with that, we're quickly just going to ask you guys to do this. Relaunch. There we go. How much you guys understand transcriptomic data? This was a really brief overview. Thank you, Rachel. But we're curious to see if it would help you guys to understand if there should be more transcriptomic talks or webinars. If you're watching this on YouTube, survey link is in the description, has all the questions, so you get a little sneak peek. But I will leave that up. And while people quickly fill out, just a quick multiple choice, we'll move on to the next part. We can leave that up for maybe 30 seconds. Yep, I think we're good. Let's keep going.
All right, so that was really just a crash course into transcriptomic data, very, very brief. But I just want to give a foundation of the type of information that is found in the ABC Atlas, since the ABC Atlas is a transcriptomic atlas. And so not only in the atlas do we have the single cell data, which I just talked about previously, where we get to maintain all of our individual cellular identities and we get to maintain our fruit salad over here. There was also another technique done called spatial transcriptomics, which not only keeps our identity of our individual fruits, but it also keeps their spatial organization, much like this fruit pie down here. And we can see the actual location in the tissue slice, right? Because with single cell sequencing, all the cells end up getting mixed together and you lose where they were in that tissue section. You can do micro dissections of really small anatomical areas to say, oh, this is from this area of the brain, but the actual spatial relationship between those cells is lost with single cell sequencing. But with spatial transcriptomics, using a technique called Marefish, or multiplexed error robust fluorescent in situ hybridization, we can actually maintain the spatial relationship of these cells, and we can actually view them in the intact tissue section. And so both of these techniques were used to create this atlas. So there were over 4 million cells profiled from over 300 different mouse brains for the single cell data. And then 400 million cells were also profiled from a few mouse brains for the spatial transcriptomic data as well. And then these data sets, these were integrated together so that we could really map out and create a full taxonomy of the whole mouse brain that also includes spatial data and spatial relationships. And so what do I mean by taxonomy? If you've seen some of our previous webinars, we had a webinar a few months back focusing on what a taxonomy is and how it's created. But the transcriptomic data from these data sets was used to create a hierarchical taxonomy of these cell types identified in the whole mouse brain. So we have our highest levels of organization. Those were called neighborhoods. There were 7 neighborhoods identified. Then below that, we had classes. There were 34 classes. Below that, we had subclasses, supertypes. And then at the very end, at the very bottom of our hierarchy, we had over 5,000 clusters of cell types in the mouse brain that were identified using these techniques. And it's a lot of data, so the ABC Atlas was created so that it's easier to access this data and sort through it yourself. And so the 3 taxonomies that are currently in the ABC Atlas include the whole mouse brain, which is a lot of the techniques I used for previously just described were used for that. We have whole human brain data, and then we also have a human Alzheimer's taxonomy as well, specifically featuring samples from the MTG and the dorsolateral prefrontal cortex. And that one also includes both single nucleus data and MERFISH spatial data. So here's the whole mouse brain taxonomy. This was published last year in 2023. This is a dendrogram to show those different hierarchies of the taxonomy that I just mentioned. So at the very top of this dendrogram, we have 7 neighborhoods listed. Then we have our 34 classes. Then we have 338 subclasses. And then the next levels we don't have on there because it goes down to 5,000. But here are UMaps, as I mentioned before, previous slides back. When you have a cell by gene matrix, that's very high dimensional data. And all that high dimensional data has now been reduced down to 2 dimensions here, into X and Y. And that's what this plot is shown here. This is a UMap, which shows all of our high dimensional data grouped together in 2 dimensions. And by 2 dimensions, I mean that dots that are closer together in this plot mean that they are much more genetically similar, and dots that are farther away are much more genetically distant from each other. And we can color code the plots. This is the same plot shown 4 different times, but color coded to show the different divisions, our classes, our subclasses, our supertypes, and our clusters. So in the plot all the way on with our 5,000 clusters here, there are 5,000 different colors corresponding to each cluster. So this is the whole mouse brain taxonomy. And there are 3 different components to this taxonomy. There's the single cell data, which is up here at the top, which was done using 10x technology. Over 30,000 genes were profiled. Here is the paper. It's in Yao et al. 2023. And then we had 2 different MERFISH data sets that also are part of this taxonomy. One is also from the Allen Institute, which was through Honghui Zhang here. This had 500 genes paneled. And then we also have another MERFISH data set from Xiaowei Zhuang from Harvard, where there were over 1,000 genes in this data set, and also coronal and sagittal here. But these are all in the same taxonomy. So if you go through any of these data sets, you'll see your 7 neighborhoods, you'll see the 34 classes, the 338 subclasses, so many supertypes and clusters. So they were organized and clustered together in the same taxonomy. But these are just 3 different data sets that you can view in the ABC Atlas to look at these data. And these were also published in a big Nature package, which is highlighted down here. These are 2 papers that were part of a 10-paper package if you're interested in learning more about that. Here's how you access that data. So this is on our ABC Atlas landing page here. This is the link that I'm highlighting in blue. These buttons here show you this to access the data. Here we have a paragraph describing the data. This is the data from the Allen Institute. So we have both our 10x data and our MERFISH data. And then we also have a few use cases here down through this green button if you're interested in how this data might be useful for designing an experiment. So we have some examples there. Here is how you can access the Zhuang data from the MERFISH data from Xiaowei Zhuang at Harvard University. Again, this is a different data set, but it is part of that same taxonomy here. So you'll see that it's color-coded in the same way. It has the same labels for the classes and subclasses. And here's how you can enter that data here. And this data set has 4 different brains. Some are sagittal, some are coronal, and over 1,000 genes. So that's a summary of the mouse brain data. We also have whole human brain data. And the whole human brain data here was just published last year in 2023 in the paper Slutty et al. This was also part of a major paper package that was published in Science. And here's a dendrogram showing that this is a taxonomy that was made from single nucleus data from human. And much like our previous taxonomy, it's divided into different hierarchical levels. But this time, our hierarchical levels have different names. In this taxonomy, they are called superclusters, clusters, and subclusters, which is different from our mouse taxonomy, which had class, subclass, supertype, and cluster. So depending on which taxonomy you're looking at in the ABC Atlas, the different levels of the hierarchy will have different names. And that's just based on the original data. And so in this whole human brain taxonomy, we have 31 superclusters, which is our highest level. And then we have 463 clusters below that. And then below that, we have 3,000 subclusters. And this was created with over 300 million nuclei from over 100 different anatomical sections. And here's more details about that data set. So in the ABC Atlas, this whole human brain is split between neuronal data and non-neuronal data. And both of these data sets came from 3 whole human brains from 3 donors, and also primary motor cortex from a 4th donor. There were over 50,000 genes profiled in both of these. And if you have any more questions, or if you want to read more about the techniques or more details about this, the paper is published. You can find that information. But all the data from that paper is in the ABC Atlas. And here's how you can access it. So again, this is on our landing page here. The link down in blue is how you can access this landing page. To explore this data set in the ABC Atlas, we have our blue button here. And then we also created some different use cases down in green to show different examples of how you might use the ABC Atlas to further your own research questions or just increase your knowledge about a certain gene in a certain area. So if you see these atlases and you're not entirely sure how they might be useful or what the different features are, I would recommend looking at these different use cases, which will walk you through how to do different steps and how to answer certain questions in the ABC Atlas. And our third taxonomy here features our human Alzheimer's data from the CAD project. And one of the authors of this paper, Kyle, actually spoke at a previous webinar that we had on January 31st. The webinar was titled, What is a Taxonomy? So if you have more specific questions about this particular taxonomy, I would recommend going back on our YouTube channel and looking at that webinar to see, to hear what Kyle had to say. And so this Alzheimer's taxonomy, again, is also split into a different hierarchy. So it's not the whole brain like we just had the whole human brain. This is specifically looking at the middle temporal gyrus and the dorsolateral prefrontal cortex. And so from these data, there were 3 classes, 24 subclasses, and 139 supertypes. And this is currently, the paper is currently online on bioRxiv if you want to read more details about how this taxonomy was created. And so what's really interesting and unique about this particular taxonomy is that there were supertypes that were seen in the severe Alzheimer's states that were not seen in the non-diseased samples. So this cohort spanned 84 donors across the entire severity of disease. And so there were the cell types that were unique in the severe Alzheimer's donors are labeled here in this taxonomy. They have this CAD marking here, which I highlight with this red arrow. So this was an interesting case where we actually saw new supertypes created that did not exist in our taxonomy in the non-Alzheimer's patients. But in our severe Alzheimer's patients, there were changes and differences in the cell populations. And so this is a huge project that involves a lot of different people from the University of Washington, from the Allen Institute, from Kaiser Permanente, from the NIH. And here are just some of the people who are involved with this Alzheimer's project. And so in the ABC Atlas, we have both single nucleus data and MERFISH spatial transcriptomic data from these Alzheimer's donors. So in the single nucleus data, we have information, we have cells from 84 donors. And then in our MERFISH data as well, we had 140 genes profiled from 24 different donors. And the CAD taxonomy in the ABC Atlas has a lot more different filters and functions compared to the other two taxonomies, because there were also clinical scores done on the donors before they passed away. So different, cognitive status, APOE status, education, there's a lot of different clinical information that you can filter by and sort through in the ABC Atlas. And here's our landing page, which describes what this data shows, where it came from, and if you want to access it, here it is here.
Awesome. Thank you, Rachel. I'm going to launch the next one. I'm curious what you guys is the data set you guys are most interested in. Again, if you're watching this on YouTube, the survey with all these questions is linked out. And while you guys fill that out, we had a question on YouTube, actually. So we had a question is, do you plan to expand your spatial transcriptomic data by adding sequencing-based spatial transcriptomic data? Also, do you plan on adding single nucleus RNA-seq data from the non-human grade 8 brains to the ABC Atlas?
Yeah, there are more data sets being added to the ABC Atlas in the future. I cannot give exact estimates on when they'll be added in which particular data sets, but there are a number of other projects going on at the Allen Institute, yes, that involve spatial transcriptomic data that in the future, will likely be added to the ABC Atlas.
Awesome. While people fill out on is which data set is most interested to them in the ABC Atlas, we'll let a couple people more fill that out. But while we check that up, should we pass it on? I'll leave it up for a few more seconds as people are still trickling answers. Again, if you're watching on YouTube, same poll is on this survey. If you're even watching a recording of this, assume you're watching a recording in the next couple weeks of May 2024, that survey will still be live. And it definitely helps us figure out what resources and more effort should we push based off these results. Great. I think we're good to move.
All right. Okay, so next I will be passing presentation off to.
Hi, my name is Alicia Fiabani. I'm a product manager here at the Allen Institute, and I'm going to talk to you a little bit about ABC Atlas, the tool. Go on to the next slide, please. So what is it? You've heard a lot about the data that's included that we've been able to show using the ABC Atlas, but I wanted to give you a little bit of a background of why we created it and what it's good for. So part of why we decided that this was necessary, there's lots of portals out there for viewing specific data sets. What we saw was a need was to show whole brain data. Data sets are getting larger and larger. The amount of data out there is getting bigger and bigger. And there really is not a way to compare data sets in the same place. And so exploring and comparing whole brain data is something that ABC Atlas is particularly good for and was designed to do. It is flexible, it is data-driven, meaning that as we add new data sets, it can expand to show all of the features and metadata that's associated with the cells that are being shown. We have a scalable infrastructure that can expand to billions of cells if we need it to. It has the ability to show, at the moment, 4 separate data sets side by side. You can filter each one of them separately by gene expression, anatomy, cell type classification, and really get a sense for how these data sets are the same and different. We also envision a platform that encourages collaboration. We highlighted the ability to share insights, to connect research and researchers across neuroscience. we've included the ability to create interactive deep links for sharing and citation. And you can also download high-res images. Excuse me. There's also links to full data sets to do deeper analysis. Next slide. So how do you get there? So you've seen this link. You can go to the landing page and it has all of the data sets that are available with descriptions and links to get there. Next. Excuse me. How to use the ABC Atlas. So once you get there, the easiest thing to do is just start exploring. So you'll see on the left-hand side, we have navigation. So you can see the top link in the left will get you to different data sets. The middle link with the circles will get you to view all of the cell properties. And you'll also be able to see genes in that little icon that looks like a DNA. You'll also be able to see cell properties and anatomical annotations using the tooltips. Filter and color by cell properties and genes. And note, not all genes are available in all data sets. For comparing views, you can compare up to 4 frames of the same or different data side by side. You can also sync your camera between views of the same data, so you can hone in on exactly the same cells. Next. Finally, as I mentioned, sharing and citation tools. You'll see the info icon scattered about the application. That will show you more information about each data set and visualization. You can find links to downloading resources and easily copy a link using the share button in the bottom left, or copy a citation properly formatted to your clipboard for easy sharing there. We'll get into more detail about learning more using our user guide and community forum later in the presentation. Excuse me, what's coming next? So this tool is meant to grow and expand as the science grows and expands. We had a question about whether we expect to add more data. Yes, absolutely, yes. As we have more data coming out of our institute, that's one of the things that we excel at is producing large data sets with novel experiments. That data can go right into ABC Atlas and be explorable. That includes more species, more modalities, more developmental stages, and more data from diverse projects. We're also exploring using generative AI to make the tools more accessible, and a lot of community outreach initiatives by this group that's presenting this webinar today. We also want to connect it to the broader ecosystem of tools. So there are so many scientists and developers creating tools for visualizing data. We want it to be interoperable with those tools. We're also building our own brain knowledge platform that will support the underlying data, making that accessible and enabling easier comparison across projects. And with that, I'll hand it over to Rachel to show you a few examples.
But before we kick on Rachel, we got a quick question on Zoom that I think would be a great time to answer now. Is there a legend defining the different abbreviations used to describe the different regions?
Yes, there is. I'm actually going to link to that in a few other ones, but the anatomical abbreviations come from the Allen Brain Reference Atlas. And yeah, if you're not familiar with those anatomical abbreviations, then I would recommend side by side having the Allen Brain Reference Atlas up on your screen or on a screen while you're also looking at the ABC Atlas. I can show you how to do that in the next couple slides, because that's something that, yeah, I personally use different acronyms for different areas of the brain, and then I learn other ones. So there's sometimes it's tricky to switch between the two sometimes, but we have ways to do it. So here's an example of a use case of How could the ABC Atlas be helpful for my own research and for my own data? So an example is, let's say you're a researcher and you're interested in studying different genes involved in opioid signaling, and you specifically want to see how different genes with opioid signaling affect sensory processing. How might the ABC Atlas help you out to figure out how you should design an experiment? So if you go to the ABC Atlas and you're looking at the spatial data here, the MERFISH data, this example is showing you how can you filter things down first by the anatomical division. So I mentioned in this use case that the researcher is interested in sensory processing. So they specifically only want to look at the somatosensory cortex. So if you go up here and step 1, click on the cell properties tab here, which our first arrow is pointing to, that will lead you to a number of different filters down here where you can filter by class, neurotransmitter type, anatomical division, and other areas. So if we go down to the anatomical division here, and again, in the Allen Reference Atlas, somatosensory cortex is referred to as SSP, not S1. So this is an example where it would be helpful to have that Allen Institute Reference Atlas side by side up so you can see what these acronyms mean. And here's a link to that reference atlas here, mouse.brain-map.org/static/atlas. That will take you to our reference atlases, both coronal and sagittal for the mouse brain, and we also have one for human brain as well. But here in the ABC Atlas, you can go and filter just by anatomical division. So here in the section, I typed in SSP under the isocortex, and then I clicked on this little ink drop here, step 4, which then filters and colors my brain sections just by that anatomical region. And if you're familiar with our reference atlas and the color code that it has, that same color code is being used here. So you see that the isocortex is green, just like the green isocortex in the reference atlas. So that's one way first to just divide by and filter by anatomical location. So the next step for this researcher is, okay, I'm looking at the somatosensory cortex, but now I want to look at, again, genes related to opioid signaling in the somatosensory cortex. So here, if they go up to the gene panel here, which is represented by this helix that we have, they click here, they can type in different genes. I search PDYN or prodynorphin. And then if the gene is in the data set, then it'll appear down here, and you can color your MERFISH cells based on their gene expression. So here in this image, we are now isolated to just the somatosensory cortex, and I've color-coded all the cells based on their prodynorphin expression. So darker color here means higher expression, lighter color means lower expression. And as I mentioned in the beginning slides, we have 2 different mouse MERFISH data sets here. We have one from the Allen Institute, and we have one from Xiaowei Zhuang's lab at Harvard. And these 2 MERFISH data sets had different numbers of genes profiled in them. So in the Allen Institute data here, there were 500 genes. In the Zhuang data, there were over 1,000 genes. So first, if you look at the Allen Institute data set and your gene of interest is not in this data set, I would then go look at the Zhuang data, because there have been times where I'm looking for genes and it's in one data set but not the other. And to get a full list of the genes from the MERFISH experiments from these data sets, these are both available in the supplemental tables, supplemental table 6 from Yao et al. And supplemental table 1 from Zhang et al. For the corresponding data sets. We also have some of these supplemental tables linked in our resource catalog, which is linked here to the left in blue. And Lauren will talk more about the research catalog at the end of this webinar. But again, here's how, if you go to the gene panel, you can filter by gene. You can also turn this whole thing into dark mode. And when it's in dark mode, the background will be black. And at that point, the coloring of the genes will switch and be opposite. So if you're in light mode, darker color means higher expression. And so because this is a taxonomy, we're not just looking at which genes were in which cells. We can also look at different classes and subclasses of cells within that hierarchical relationship and see how they are involved with the genes. And so here, Now I've gone to the cell properties tab, and instead of clicking on the anatomical division, now I'm clicking on class. And I can go through and filter by different classes. So remember, in this taxonomy, we have 34 different classes of cells. And so in this image here, I currently have the cortex MGE GABAergic cell class highlighted. So right now, all the cells that are being shown here are being filtered to just those GABAergic MGE cells, and they're color-coded based on the prodynorphin expression. And if you're confused or have some questions on what these acronyms mean, these labels of these classes and subclasses, and what CTX-MGE mean, there's an annotation table that was published with this data set, with this paper, which is supplemental table 7 from Yao et al. We also have this table linked here on the website. It's the cell annotation table, and that will show you, give you a bit more information on the namings of these classes, and more information on how they were divided up. And then what's our next step below classes? Below class, we have our subclasses, right? We had 34 classes of cells, which then split into 338 subclasses of cells. So I can go further and filter further by going down that hierarchy. So within the cortex MGE GABAergic class, I specifically now have filtered down just to the SST GABAergic subclass of cells. And by doing that, if we flip between the back, we can see that the darkest cells in my class are in this particular subclass. So that's new information for me. That's showing that the prodynorphin, the gene that I'm looking at, it's not equally expressed in my MGE GABAergic interneurons. It's actually, the darkest levels are really within my somatostatin groups. That's new information for me and can help me design my experiments. And again, if I'm confused on the namings of the subclass, I can go back to that cell annotation table to see. And there's also an annotation table like this for the whole human brain data as well, from the data for Saletti et al. And that's also linked on our resource catalog. So now, based on this information using the ABC Atlas, I can see that prodynorphin is not only highly expressed in MGE-derived GABAergic cells, it's actually specifically within the somatostatin class. And that's interesting for me if I want to look at opioid signaling in the somatosensory cortex. So now that I have this information, I can continue designing my experiments based on different mouse models and genetic tools that are available. And the Allen Institute has created a number of different transgenic mouse lines, AAV tools, enhancer viral tools, other tools for use. And those are all linked here, portal.brain-map.org/genetic-tools. And if you go here, you can find the list of the transgenic lines and different viral tools that can help you plan and design your experiments. All right, and that is the end of this use case. And here are some other use cases briefly that the ABC Atlas is really great for, such questions as, are genes X and Y co-expressed in the same cell type? In that previous example, I only showed one gene, but you can actually have multiple genes and flip between the two different genes and see, are they both highly expressed in the same cell? Where is gene X expressed in the brain? So in my use case, I just filtered my cells to the somatosensory cortex. But if you want to look at the whole brain, you can just look at the genes in the whole brain, figure out where they're expressed. Is gene X expressed in a specific cell type? That's a question I was just answering. Is prodynorphin equally expressed in MGE interneurons, or is it in a specific group? Where is cell type X in the brain? If you know a particular cell type that you're studying and you want to see where else it is present, you can use the ABC Atlas to answer that question. What's the spatial relationship between cell type X and cell type Y? Because there's Mirfish data, spatial transcriptomic data in this data set, and not just single cell data, we can start to answer some of those questions, right? We can see, if a cell is really in upper layer 2-3 of the cortex versus lower layer 2-3. Is it closer to this other cell type? You can really start to pick apart those spatial relationships. Is cell type X more prevalent in region Y versus region Z? Because this is a whole brain taxonomy, you can start to answer those questions and look at, where the cell types are, where they're most expressed, and see how they differ between different anatomical regions. Is this subclass more diverse than another subclass? So because our subclasses are then divided into supertypes and then clusters, you can see which subclasses have many more cell types within them and which ones do not. So you can look at the diversity of different subclasses as well. And there are some questions that, as of right now, cannot be answered in the ABC Atlas. So just to clarify, the data here that's shown, the MERFISH data, is based on cell segmentation based on the somas of the individual cells. So if you're looking for questions like, I'm interested in cell-to-cell contacts, I'm less interested in synapses, that type of information is not shown in the MERFISH data and cannot be answered in the ABC Atlas. So any type of synaptic, if you're looking for any information about dendrites or axon terminals, you will not find that information here with the MERFISH data. What's also shown here is the centroids of the cells. So when you zoom in and out on the ABC Atlas, the shapes, the dots of these cells will change size based on your zoom. And that's because what's shown is the centroids of the cells, not the cell shape or size. So again, you cannot use this MERFISH data, that you cannot use the data in the ABC Atlas to say, well, is this soma, this close to this soma, or is it this size or shape? That type of information right now is not there in the ABC Atlas.
Great, thank you. And those are some awesome use cases, and we would love to hear what other ways you guys can imagine using ABC Atlas. Maybe Rachel, you can go back a slide to leave that up, but we definitely want to hear from you guys. What are ways you can envision using the ABC Atlas that helps us make sure that use case is supportive? We have resources for that. So I'll leave that open, and it's okay if you don't know at this point, but we're just trying to gauge what other ideas people have. Since it's free response, we'll leave it up a bit more. And actually, I'm going to take one of the questions we had from the YouTube chat, because I don't think I'll be talking about it. But is there a way to access the FASTQ files for more granular analysis for ABC Atlas?
Yeah, so there are a number of Jupyter notebooks that were made to access the raw data for the whole human brain and the whole mouse brain data sets. And those should be linked in our resource guide that we have. But yeah, a number of Jupyter notebooks were created to walk through if you're interested in just downloading the raw data yourself, doing your own analysis, and those are written in Python. So if you're comfortable using Python, that is one way to access those data. And they're also available, if you don't want to use the notebooks, all of that data is also available online at the Allen Institute data catalog as well. If you go to the project summaries of these particular data sets, you can find another way to access that data as well. And if you have any questions about accessing the data or getting lost on the website, feel free to reach out, post on our community forum, or send an email. We're here to help out and help people find this information.
And I'll just address another question that was mentioned earlier. Do you have patch-seq data? We do not currently have patch-seq data in the ABC Atlas, but I did. In the chat, we linked out to our human patch-seq data set, and also we had a previous webinar focused on patch-seq. It's called the Techniques Behind Cell Typology Explorer, which talks a bit about just patch-seq in general. So linked out to that. Also then, how do we do this? Yeah, so that is a short answer. I guess I can move on. I'll leave that up for a bit and just briefly go over, in the interest of time, some other resources we have to support for ABC Atlas. Again, if you're watching this on YouTube, we have that survey in the description with all these questions. So as mentioned, Rachel kind of teased it. We actually have a huge new catalog called Cell Type Support, which helps you figure out all the resources we've created to help your use of ABC Atlas, but also any other cell types work. So there's 3 sections of this. It's a what is, how to, putting together. This is all found on our brain map. This is the URL, help in community /guide cell types. But if you go to either the cell types main page or the help in community page, you'll see this title with the little magnifying icon. So there's 3 sections, and I'll kind of review what those 3 sections are about. So yeah, the what is a great kind of introductory. If you're a little bit new to cell types or just want a refresher, we have a lot of our links to our great resource of that, such as Hunkwey's review paper on what is a cell types and how to define it. We have a brief one on what is transcriptomics. We link out to previous webinars, such as like our cell type taxonomy webinar, a great guide to what is a UMAP, and also a guide to our what is a patch seek. So this is a great introductory if you're a little bit new to cell types and just need to get some grounding. Slide. Then we have next whole section called how to. So this is a lot of our more technical support. So this includes our protocols that are freely available, our guides, a lot of the supplementary annotation tables that Rachel mentioned will also be found on this section. So this includes the cell type nomenclature, patch-seq, our RNA-seq protocols, our patch-seq protocols, the software we use for morphology, our cell type knowledge explorer. So if you attended any of these previous webinars, we've done a lot about cell types knowledge explorer. Some of our use cases and other support can be found there, along with the ABC Atlas. So a lot of the annotation tables, again, Rachel mentioned, is found on the ABC Atlas, is found on this support guide as an easy way for you to pull it if you don't want to dig through all the supplemental tables we have. Also, we have the guides from AL to ABC Atlas. After this webinar, the link to this webinar will also be there. So if you want to quickly find this webinar, it will be there on that guide. Next slide. And finally, we made this kind of master key table. It's kind of confusing to figure out, okay, what is the research subject? What tool does that go to? What taxonomy does that go to? What paper does that go to? So we have a whole table that's linking all those resources as an easy way for you to find what taxonomy went with that tool, what paper was that tool from. So if you're familiar with the paper, you can search from the paper and see what tool, if we have a tool on it. If you're familiar from the tool side, but you don't know where the taxonomy is, this table is there for you. Yeah, and as mentioned, we're continuing building more resources to help your use with ABC Atlas and beyond. Some thoughts we have for making one is have a more in-depth transcriptomic introduction. Rachel did a really great job this as a quick teaser, but we can definitely go a lot more in depth about, spatial single cell versus single nucleus. We've also thought about having more dedicated user guides for each data set, from the human, the Alzheimer's, the mouse whole brain. More use case examples. I think Rachel's a great use case examples, and we have a handful of but if that is something people are interested in, we're definitely eager to hear more if those are useful for you guys, along with virtual office hours. We've been thinking about having that. So with that, I kind of just want to ask you guys if there is a particular resource you guys are interested in using at for your ABC Atlas look. So I'll just quickly run that poll on that. On if there's a particular resource you think we should help. This really helps us figuring out if we should start making that resource. Again, if you're watching the survey, if you're watching it on YouTube, the survey is on the description link. And then while I leave that up, we had a previous question, was there any way to query the data by R? I'm not sure, Rachel, if we have one.
Yeah, we have a couple people at the Institute who do their R's in Python and a couple in R. We have some R notebooks for Map My Cells, and Map My Cells is a tool for using these taxonomies featured in the ABC Atlas to map your own data to it. But right now, I believe for accessing the data directly from ABC Atlas, I believe they're all in Python, but that's something that's useful for us to know. If the same notebooks would be helpful if they're created in R, that's something we can pass along to our team and say that, there's a demand for that. That would be useful. But yes, right now some things are either in Python or R, but we have people here who would likely be able to help out with that. Yeah. Or if there's or we could ask in our forum, our community forum, if you have specific questions there as well. Sometimes, the people who make the notebooks can directly answer and provide feedback there as well.
As Rachel mentioned, we're really interested in what resources you guys would like. So, this poll helps. Yeah, someone says please implement them in R. We'll pass that along, but also, as we mentioned, I think previous webinars, community forum is super active. I know the people who write in R watch that. So if you make your ask needs there, we'll hear that well. That's a great way to kind of mention what you want. But also, we're also at SFN, so I guess that'll be a great segue to the next couple slides. Rachel, if you want to move on, next slides. Yeah, so as we mentioned, this has been a series, and this is the last in the six of the series. All the previous webinars from Cell Types 101 to Cross Species to Map My Cells is all on our YouTube channel. But as of May 24, we'll also be at FENS, AIC, and SFN. So if you have some more questions, we'll be there. As long as a quick sneak preview, we will have a satellite workshop at SFN. It's usually that's right before SFN starts. It will probably be focused on ABC Atlas. And so if you have a lot more questions about what resources you would like, that is the time to voice them. And with that, I'm going to launch, I promise, the last poll of just asking people if they've used ABC Atlas. This really helps us gauge if this webinar was helpful for you guys' interest on that. But with that, I can also take any other questions we have. We have a couple on the chat and I will address them. How do we can we access the merfish images?
As of right now, the raw fish, yeah, images are not available, but that's, again, something that, yeah, we can talk with the spatial transcriptomics team about how to access that data. But I believe right now, yeah, those are not publicly accessible.
Here you go. Yeah. Yeah, if anyone else has any other questions in the chat, do that here on that. I'll leave that up. Again, thank you for everyone for attending this webinar and everyone who's attended the previous webinars. This is kind of a new one we've done with a whole series of health cell types. So hopefully it was helpful for you guys. It was helpful for us. But yeah, thank you for everyone for joining. I'll keep it open for any more questions if people have them. Oh, do you have plans to expand the ABC Atlas to the brainstem/medulla?
Yeah, so there in the human data set, there is a small amount of, yeah, some spinal cord data and some brainstem data as well. I think in the, yeah, again, that's in the whole human data, the SLEDI data set. There's a very small amount there. In the human, yeah, in the mouse brain, I don't believe there is right now. And yeah, I'm not aware of any immediate plans to add that, but that's helpful feedback to know. That would be useful.
Another question, is there any help with cell segmentation tools and also a way to co-register your own images from merfish to yours?
Yeah, I know that cell segmentation is something that's challenging, and it's definitely going constant reevaluation here with the spatial transcriptomics team. I don't know if we have any public resources. I know that because the VisGen is what's used for this merfish data, and I know that they have, VisGen comes with its own segmentation algorithm. So I've been to the VisGen website if you're using that version to look at how they do their cell segmentation. But as of right now, the one that we have for the Allen Institute, I'll have to check if it's on our GitHub, because we do have a lot of information on our GitHub, but I do not believe that the cell segmentation, because that's still, I think, in the works here or being improved. I do not believe that is currently on our GitHub. But I would check, yeah, the VisGen website I know has support resource if that's the platform you're using for your merfish for the segmentation. But if not, that'd be helpful to know for a future webinar if we just want a webinar on cell segmentation, because right now we're planning what topics we want to cover for the next 12 months of webinars. And so if that's something that'd be very useful for the community, we could definitely consider that and just have a whole webinar focused just on cell segmentation.
How can we access office hours? We are still trying to decide how to do office hours. So if that is something you're interested in, this question came from the YouTube chat, put that in the Survey Monkey. That's one of the reasons why we have this chat. If we see a lot of people are interested in office hours, obviously that takes a lot of dedicated time from our scientists. So we want to make sure that the office is filled. But so if that's something that, and if it's something we'll do, we'll make sure we'll announce it in our social medias, or if you're part of our newsletter, part of our mailing list, our events page, that's how you can hear about it. But if you attend SFN as well, we'll definitely have some sort of office hours there as well. Awesome. Great. I don't see any more questions coming in. Thank you for everyone attending and filling out all these questions. Again, it really helps us figure out what resources we're building. As Rachel said, we're kind of in that planning phases of what resources we are going to build next, especially with ABC Atlas. So your feedback, super helpful, and hopefully it helps you with the resources you can do with your science. With that, well, thank you again, everyone, for attending, and I hope you guys enjoy your Wednesday.